Good morning, students. Today we are going to learn about the electron transport system or electron transport chain along with oxidative phosphorylation. We have seen photophosphorylation in photosynthesis. So in respiration, oxidative phosphorylation. So what is this electron transport system or electron transport chain? Why electrons should be transported? So from the glucose, the electrons and protons, they come out through oxidation. What is oxidation? Removal of electrons or hydrogens or addition of oxygen. Here there is no addition of oxygen. So liberation of electrons as well as protons. Those are stored in the NADH2 or FADH2. NADH2 and FADH2. So after taking those electrons, these NADH2 and FADH2, we call those as reducer coenzymes. Okay, so these NADH2 and FADH2, those are oxidized through electron transport system. Those electrons which are coming out from this NADH2 and FADH2 are accepted finally by oxygen molecule and it joined with the protons and it forms water molecule. This is the actual process. And during this process, protons are also released and those are used for the synthesis of ATP. Okay, and come to this location of electron transport system or electron transport chain. Where is this located? In the inner mitochondrial membrane. We know the mitochondria. This is the mitochondrial outer membrane and this is the inner membrane and it forms infoldings and this is the matrix. Okay, on this inner membrane. So these are the cristae. Along the cristae, this electron transport chain takes place. On the cristae, you can see stacked particles called as oxysomes or F0, F1 particles or elementary particles, we know that. So on that ATP synthesis takes place. So along the cristae, you can see electron transport and with the help of these oxysomes which are present on these cristae, ATP synthesis takes place. Okay. So where are these? In the inner mitochondrial membrane. So electron transport chain is located in the inner mitochondrial membrane. Now come to the complexes involved in electron transport system and oxidative phosphorylation. See, don't confuse. It will be very easy if you understand. Follow me. Okay. So the complexes involved in an electron transport system or electron transport chain and oxidative phosphorylation. Here 5 complexes are involved. One is complex 1. What we call this? NADH dehydrogenase complex or NADH ubiquinone oxidoreductase. Okay. So NADH dehydrogenase complex or NADH ubiquinone oxidoreductase complex. Okay, how is this? You see this one. This is the NADH dehydrogenase complex. It consists of 2 prosthetic groups. One is FMN and the one is FES. Okay, so NADH dehydrogenase complex means here NADH only involved but not FADH. So this NADH2, it is releasing 2 electrons. These 2 electrons are accepted by FMN and then those are passed to the FES. Then those are accepted by ubiquinone. So this ubiquinone is mobile electron carrier like plastoquinone. There. This is mobile electron carrier. Okay. Now, at the same time, protons are also released from this NADH2 and it will become NAD. See, I am showing here 2 protons. In our NCERT books, there 4 protons are there. For 2 electrons, 4 protons they come out. So here those 2 protons from the those are the free protons which are present in the matrix. Those are also come out into the intermembrane space, perimitochondrial space. Okay, remember this. So this is NADH2 dehydrogenase complex or NADH ubiquinone oxidoreductase complex. Oxidoreductase complex. It has two prosthetic groups, FMN, flavin mononucleotide, and iron sulfur. Okay, this is about the complex 1. And what about the complex 2? This is succinic dehydrogenase complex. Succinic dehydrogenase complex or succinate ubiquinone reductase. Succinate ubiquinone reductase. This FADH2 releasing 2 electrons. Those are passed through FES and then to the ubiquinone pole. So this ubiquinone is undergoing, so the reduced ubiquinone is called as ubiquinol. UQH2 means ubiquinol. What is this ubiquinol? Reduced ubiquinone. Okay. Next, complex 3. Complex 3 is called as cytochrome C reductase or UQH2 cytochrome C reductase. So this cytochrome C reductase is having cytochrome B, FES and cytochrome C1. Cytochrome B, FES and cytochrome C1. So this cytochrome C1, it releases those electrons to the cytochrome C. What is the cytochrome C? Like ubiquinone, this is also mobile electron carrier. Mobile electron carrier. Then complex 4, we call this as cytochrome oxidase or cytochrome C oxidase. What it has? It consists of cytochrome A1, cytochrome A3, and the cytochrome A3 is having 2 copper centers, copper A, copper B, or copper 1 and copper 2. Copper 1 and copper 2. So from this copper B, these 2 electrons, finally those are accepted by half molecule of oxygen. Okay. Finally, what is the 5th complex? We call this as ATP synthase complex or F0F1 particle or Fernandez-Moran subunit or oxysome. These are all the names for the complex 5. All are same only. Okay. It consists of F0 and F1. So this F0 is embedded in the mitochondrial membrane. In a mitochondrial membrane, and this F1 is protruding into the matrix of mitochondria. Okay. So I said this ubiquinone. No, what is this ubiquinone or coenzyme Q? It is a mobile electron carrier. What is ubiquinol? It is reduced ubiquinone. Where is this ubiquinone present? In between 1st and 3rd complexes and 3rd and 2nd complex. I'll show. I'm showing that. So where is this ubiquinone? In between 1st complex and 3rd complex, 3rd complex and 2nd complex. The arrangement of all these complexes: 1st complex, 3rd complex, 2nd complex, 4th complex, 5th complex. Like that those are arranged. And cytochrome C, it is a mobile electron carrier in the outer surface of the inner mitochondrial membrane. Outer surface of the inner mitochondrial membrane. If this is the inner mitochondrial member, here this cytochrome C is present. Okay. So this complex 1, 2, 3, 4, those are for electron transport. Complex 1, 2, 3, those are for proton transport. They act as proton pumps. Okay. When you see this complex 5, this is the structure of the complex 5, ATP synthase complex. Okay. For every 2 protons, 1 ATP is synthesized. Okay. And what is it? This is the F0 of the ATP synthase complex. So this facilitated diffusion channel, this one means the protons, they move from high concentration to low concentration along the concentration gradient without using energy. We call that as facilitated diffusion. We know that. So this is facilitating. It is showing facilitated diffusion. So this is facilitated diffusion channel. It is the integral membrane protein complex. And what about this? This is peripheral membrane protein complex. So this is the site for the synthesis of ATP. Site for the synthesis of ATP. So these are all the complexes. How they are arranged, I'll show. Before that, we should know the number of NADH2 and FADH2 formed during aerobic respiration. We know that in glycolysis, one oxidation reaction. Where is that? Glyceraldehyde 3-phosphate to 1,3-bipga. One oxidation reaction. So 2 NADH2 are formed. Then next come to the link reaction or intermediate step. There pyruvic acid. From pyruvic acid is undergoing oxidation, decarboxylation and condensation. As a result of that, acetyl coenzyme is formed and 2 NADH2 are formed and 2 carbon dioxide are also formed. No direct ATP there. Okay. Pyruvic acid is undergoing oxidative decarboxylation and condensation. So that a 2 carbon acetyl coenzyme is formed and 2 NADH2 are formed. In glycolysis, 2 NADH2. Link reaction, 2 NADH2. Okay. And in Krebs cycle, isocitric acid to oxalosuccinic acid, 1 oxidation. So 2 NADH2. Alpha ketoglutaric acid, 5 carbon compound to succinyl coenzyme, a 4 carbon compound, 1 oxidation, 2 NADH2 are formed. Malic acid to oxaloacetic acid, 1 oxidation, 2 NADH2 are formed. Total NADH2 till now, glycolysis 2, link reaction 2, and in Krebs cycle 6. So total 10 NADH2 are formed. And what about FADH2? Succinic acid to fumaric acid. 2 FADH2 are formed. Okay. Total 10 NADH2 and 2 FADH2. So all these they are entering in, all these 12 NADH2 reduced coenzymes, NADH2 and FADH2, they are entering into the electron transport chain or electron transport system for oxidation. Okay. So now you see now how they are arranged. You think that this is the mitochondrion. Look here. This is a perimitochondrial space. What is this perimitochondrial space? Space present in between the two membranes. And this is the mitochondrial inner membrane. And this is the matrix. Okay. Now in the matrix, we are seeing the, for one NADH2 we are seeing. For NADH2 only you see. Listen carefully. Not the FADH2. We are seeing about NADH2, not FADH2. For NADH2, oxidation means first complex is involved. Third complex, this is the third complex. Third complex is involved. Fourth complex is involving. And fifth complex is also involved. Okay. So, first complex, third complex, fourth complex, and for ATP synthesis, fifth complex. These are only involving, but not the second one, because second one is FADH2, succinic dehydrogenase complex, that one. FADH2 there. So, we are going to see about NADH2 only. That how many NADH2 are formed? 10 NADH2 are formed. Now we are seeing about 1 NADH2. Okay. So this NADH2 is undergoing oxidation and it is releasing 2 electrons from here. At the same time, 4 protons are also going out from the NADH2 and from matrix into the perimitochondrial space. But we have to count 2 protons. See, according to the NCERT diagram, 4 protons. According to the NCERT matter, 2 protons, 1 ATP. Remember this. Understand this. So, out of the 4 protons, 2 protons, those are free protons from the matrix, they are coming. From NADH2, 2 protons are only coming. Okay, and these 2 electrons are accepted by FMN. Okay, and then to FES. These 2 electrons are passed to the ubiquinone. Ubiquinone. It is reduced to ubiquinol. Ubiquinol. Got it till here? So, from one NADH to 2 protons and 2 electrons are released. These 2 electrons are passed through FMN, FES and reached to ubiquinone. And it is reduced to ubiquinol. Reduced to ubiquinol. Okay, in the intermembrane space, 2 protons are there. According to the NCERT diagram, 4 protons only. For ATP count, we have to consider 2 protons here. Okay. Now, from ubiquinol, these 2 electrons are passed to the third complex. What is this complex? Cytochrome C reductase, a cytochrome BC1. Cytochrome BC1. It consists of cytochrome B. Third complex is having cytochrome B, FES and cytochrome C1. These are the components of the third complex. So these electrons are passed from cytochrome B to FES, FES to cytochrome C1. Okay, and from cytochrome C1 to cytochrome C. Cytochrome C1 to cytochrome C. While these electrons are passing from the matrix, again 2 protons are released. 2 protons are released. See, from the matrix 2 protons are released. Here 4 protons in the NCERT diagram. We have to count for ATP synthesis means 2 protons only. Remember. Okay. Each time 4 protons are only coming. But we should consider 2 protons for ATP synthesis. Now, where are the electrons now? In the cytochrome C. Where is the cytochrome C? What is the cytochrome C? Mobile electron carrier. Where is this present? What is that? Cytochrome C. Outer surface of the inner mitochondrial membrane. See, cytochrome C is towards the outer surface of the inner mitochondrial membrane. Outer surface. This is inner surface. Outer surface, this one. Okay. So, from cytochrome C, these electrons are passed to the fourth complex. What is fourth complex? Cytochrome oxidase. Cytochrome oxidase. What are the components present in the cytochrome oxidase? The fourth complex. Cytochrome A1, cytochrome A3. A1, A3. This A3 is having copper A and copper B. So, electron transport, you see. These are all very important. The sequence of electron transport. From cytochrome C, those are passed to copper A, then to cytochrome A1, then cytochrome A3, copper B, and finally to the half molecule of oxygen. The final electron acceptor is half molecule of oxygen. Half molecule of oxygen. And here also 4 protons are released from the matrix. 4 protons are released from the matrix, but 2 protons, they join with the half molecule of oxygen and then form 1 water molecule and 2 protons into the perimitochondrial space. 2 protons into the perimitochondrial space. Okay, this is for 1 NADH2. How many protons are present in the perimitochondrial space? We have to count these 2 protons, 2 protons, 2 protons, 6 protons. So, these 6 protons, proton gradient is established. So, these 6 protons, they move through the F0 and F1 of 5th complex, ATP synthase complex. 6 protons, they move from perimitochondrial space through the F0, F1. F0, F1 of ATP synthase complex into the matrix. Into the matrix. For 2 protons, 1 ATP. For 2 protons, 1 ATP. For 6 protons, 3 ATP. Okay. So how ATP are synthesizing here? What is this transportation? Facilitated diffusion. What is facilitated diffusion? Anything moving along the concentration gradient without using energy here. Okay, anything when it is moving along the concentration gradient means some energy is released. What is that energy? Proton motive force. That proton motive force is used for the joining of ADP, adenosine diphosphate, and inorganic phosphate, and it forms ATP. Okay, so for 2 protons, 1 ATP according to the diagram, but according to the matter in the NCERT, 4 protons is equal to 1 ATP. And finally, they have given 40 ATP only. If we count like that, we don't get actual number. So here, for 2 protons, according to the diagram only, we have to see. See, look here. Here, this is NCERT diagram only. Look here. 2 protons, 1 ATP. According to the NCERT, 2 protons, 1 ATP. Diagram wise. If it is matter wise means 4 protons, 1 ATP. According to the option, you have to answer. They don't give both. They don't confuse you. Okay. So, 6 protons means 3 ATP. For 1 NADH2, we are getting 3 ATP. Remember. And how many water molecules we are getting? 1 water molecule. For 1 NADH2, 1 water molecule. What is the final electron acceptor here? Half molecule of oxygen. And what about the proton acceptor, final proton acceptor here? Oxygen only, and it is becoming water. Okay. So, for 1 NADH2, 3 ATP. We will see later about the number of ATP. Now, come to the FADH2. This is FADH2. Okay. So, FADH2 also releasing the 2 electrons and it will become FAD. The 2 electrons are passed to FES and to the ubiquinone pole. And from ubiquinol, these electrons are passed to third complex. This is the second complex, FADH2, succinic dehydrogenase complex, that one. So, the 2 electrons from FADH2 to FES, FES to ubiquinone. So, reduced ubiquinone is ubiquinol. From ubiquinol, these electrons are passed to the third complex. So, cytochrome B, FES, cytochrome C1, and then to the cytochrome C. During this process, these 4 protons according to the diagram, and according to the matter, 2 protons. We have to count these 2 protons. Don't take this part because NADH2 is not involving in this. This is about FADH2. Okay. So, where are the electrons now? Cytochrome C. From cytochrome C to the fourth complex. Copper A, cytochrome A1, cytochrome A3, copper B, and then to the half molecule of oxygen. Okay. So, here how many protons are there in the perimitochondrial space? Here 2 protons, here 2 protons, 4 protons. Those 4 protons, they move through F0, F1 and it forms how many ATP? For 2 protons, 1 ATP we know. So, for 4 protons, 2 ATP. For FADH2, 1 FADH2 is equal to 2 ATP. For 1 NADH2 is equal to 3 ATP. Like this you should count. Okay. Now, for 1 FADH2, 2 ATP and 1 water molecule. For 1 NADH2, 3 ATP and 1 water molecule. Okay. Now, come to this. 1 NADH2, 3 ATP. How many we have? 10 NADH2. So, 30 ATP. Okay. And 1 FADH2 is equal to 2 ATP. So, 2 FADH2, we have 2 FADH2. So, 4 ATP. 4 ATP. So, through electron transport system and through oxidative phosphorylation, we are getting 34 ATP. For OP we are getting 34 ATP. Okay. And SP in glycolysis, how many substrate level phosphorylations occurred? 2. So this is OP. Okay. And SP 4 ATP through glycolysis and 2 ATP Krebs cycle. Okay. Total how many ATP now? 40 ATP. But net gain is 38 only because in glycolysis 2 ATP are utilized. So net gain of ATP 38 ATP. Okay. Now what about the inhibitors? Inhibitors of electron transport chain. Inhibitors of electron transport chain. So here during this movement, one question. So in electron transport chain, electrons move from low redox potential to high redox potential, or high redox potential to low redox potential, low redox potential to high redox potential. What is that low redox potential? What is the meaning of that? That a substance tends to lose electrons to other substance. So everywhere those are ready to release the electrons, lose the electrons. So low redox potential to high redox potential, these electrons are moving. Is this a downhill journey or uphill movement? Downhill movement only. What is the downhill movement? Downhill movement means always from high to low only. High concentration to low concentration. So the passage of electrons from one enzyme to another or the next is downhill journey with a loss of energy at each step. Low redox potential to high redox potential means it is related to electron. The substance is ready to lose the electron, low redox potential, and the substance which is ready to accept the electron, high redox potential. That is low redox potential to high redox potential, electrons are moving. Okay. So based on the energy, downhill movement, this one, downhill journey, everywhere. So high concentration to low concentration only, high energy to low energy only. Everywhere some energy is losing. It is when it is losing, every step it is losing some amount of energy. That also you should remember. Okay. Now inhibitors. What I am asking? Inhibitors. So fourth complex that is inhibited by fourth complex. The electron transport is inhibited by cyanides. So cyanide, it inhibits the complex 4 of the ETS, cyanide. Fourth complex. So it can't transport the electrons. Carbon monoxide also. Carbon monoxide and cyanides, they inhibit the electron transport in the fourth complex. Okay. Now, this is aerobic respiration, we know. What is anaerobic respiration? In the absence of oxygen. In the absence of oxygen, we see that alcoholic fermentation and lactic acid fermentation. Lactic acid fermentation. There is no oxygen at all. Yeast cell, we see alcoholic fermentation. No? So, there, which one is the final electron acceptor? In a anaerobic respiration like yeast cell, in the absence of oxygen, when this respiration is anaerobic respiration is taking place, what is the final electron acceptor? In aerobic respiration, half molecule of oxygen is the final electron acceptor, we know. Okay. In yeast, what is the final electron acceptor? We know that in so this glucose through glycolysis pyruvic acid is formed. This pyruvic acid is undergoing decarboxylation and it forms acetaldehyde. Acetaldehyde during alcoholic fermentation. Acetaldehyde is undergoing reduction and it forms ethyl alcohol. Ethyl alcohol. So here what I am asking, which one is the final electron acceptor? Acetaldehyde. Acetaldehyde. Here only reduction is taking place. It only taking the electrons from NADH to acetaldehyde. It is giving ethyl alcohol. What about lactic acid fermentation? Lactic acid fermentation. So this pyruvic acid is undergoing reduction and it forms lactic acid. No? So here this also pyruvic acid is the final electron acceptor in lactic acid fermentation. Remember, in aerobic respiration, oxygen is the final electron acceptor. In anaerobic respiration like alcoholic fermentation, acetaldehyde is the final electron acceptor. And in lactic acid fermentation, pyruvic acid is the final electron acceptor. So this is about our electron transport system.